All right, today we are diving deep into our own genetic code. We're going to peel back the layers on everything you thought you knew about how your genes work. And let me tell you, what's really going on is way more dynamic and just way more interesting than what they taught us back in high school biology. So you remember the Punnett square, right? That little grid with the big B's and the little b's that was supposed to figure out eye color and stuff like that. It all felt so neat and tidy. You get one gene from mom, one from dad, and whichever one's dominant, well, that's the one that wins. Seems simple.
But what if I told you that simple story is just like chapter 1? The reality of genetics is this sprawling epic. It's got plot twists, it's got characters with hidden abilities, and a ton of influence from the outside world. Those simple rules? They're really just the starting point for a much, much bigger story.
So, to really get how weird and wonderful this all gets, we need to make sure we're on the same page. Let's just do a super quick refresher on that classic, simple model of dominant and recessive traits.
A classic example of this is albinism. You know, it's a recessive condition that means you only actually show the trait if you inherit 2 copies of a very specific version of a gene, what we call an allele. In this case, it's the little a allele, and it's for an enzyme called tyrosinase. And that enzyme is absolutely essential for making melanin, which is the pigment that gives color to our skin, our hair, and our eyes.
So here's how it shakes out. If you have at least one of the dominant big A alleles, your body makes the enzyme, you produce melanin, everything's normal. But if you end up with 2 of the recessive little a alleles, that enzyme is inactive. No melanin gets made, and the result is the albino phenotype. Simple, right? Hold that thought.
Okay, this is where the story really starts to get good. We are now moving way beyond the simple 2-party system of one dominant and one recessive allele. The plot is definitely about to thicken.
So what happens when a gene isn't just a simple choice between A and B? What if there are multiple versions, these multiple alleles, for a single gene, all just kind of floating around in the population?
And you're already totally familiar with a perfect example of this: human blood types. For the gene that determines your blood type, there aren't 2 options, there are 3 main ones: A, B, and O. And this is where we see a new rule pop up. A and B are both dominant over O. But get this, they are co-dominant with each other. If you inherit an A and a B, you don't get one or the other, you get type AB blood. They both show up to the party.
Now let's kick it up a notch and look at cats. The gene for their fur coloration has at least 5 different alleles, and these alleles can give you everything from full, rich color to the special pattern on a Burmese or a Siamese cat, all the way to a totally white cat or a true albino.
What this does is it creates something called a dominance hierarchy. See, it's not a free-for-all. There's a really clear pecking order. The allele for full color, it's the king. It's dominant over everything else. The albino allele is way down at the bottom, recessive to all the others. And in the middle, you get these really cool relationships. For example, the Burmese and Siamese alleles, they're co-dominant with each other. It's like this whole complex chain of command, all within a single gene.
Okay, so we've got that genes can have lots of different versions, but now let's add another twist. Just because you have the gene, it doesn't mean it's actually going to show up. Think of it as the genetic version of a maybe clause. This brings us to a concept called penetrance. A great example is polydactyly. It's the trait for having extra fingers or toes. It's caused by a dominant allele, but, and this is the key, not everyone who has that allele actually grows an extra digit. Penetrance just measures the percentage of people with the gene who actually show the trait. So if only, say, 90 out of 100 people with the gene have an extra finger, we say that gene has 90% penetrance.
So the easiest way to think about penetrance is as a simple yes or no question. You've got the gene, so does the trait show up or not? It's like the light switch of genetic expression. Is it on or is it off? But if penetrance is the light switch, then expressivity, that's the dimmer dial. It's not about if the trait appears, it's about how much it appears. For those people with polydactyly who do show the trait, the expression can be all over the place. It might be a tiny little extra flap of skin, or it could be a fully formed working finger.
And getting this difference is really key. Penetrance asks yes or no. Expressivity asks, how much? And together, these two ideas explain why genetics is so much more nuanced than those simple Punnett squares ever let on.
So what's causing all this variation? Why doesn't a gene just do the same thing every time? Well, it turns out genes don't operate in some kind of vacuum. The environment that they're in can completely change the story.
A really amazing example of this is something called a temperature-sensitive allele. Just imagine an enzyme that only works if it's in that perfect Goldilocks zone. Not too hot, not too cold. If the temperature gets outside that sweet spot, the gene is still there, but the protein it's trying to make just can't do its job.
And this brings us right back to our cats. The classic look of a Siamese cat is the perfect showcase of a temperature-sensitive allele. See, the enzyme that makes dark pigment, it's inactive at the cat's warm core body temperature. That's why its body is a light color. But on its cooler parts, the ears, the paws, the tail, the temperature is just right. The enzyme switches on, and you get those dark points that Siamese cats are famous for. It's genetics and a thermostat, all rolled into one.
All right, so we've seen how even a single gene can get pretty complicated. But the final and maybe biggest piece of this whole puzzle is that most traits we see aren't the work of just one gene. They are the result of an entire ensemble cast of genes all working together.
You know, things like height or even risk for certain diseases, they're not decided by a single on-off switch. They're polygenic. That's a fancy word that just means they come from complex interactions between lots of different genes and all their alleles. Let's see how that plays out.
So let's go back to cat coats one last time. But now we're looking at two genes working together. You've got one gene for the main color, let's say black versus chocolate. And then you have a second gene that acts like a dilution switch. So when you breed cats that are mixed for both of these genes, you get this really classic 9:3 to 3:1 ratio in their genetics. This is the underlying recipe.
And now we can see how that genetic recipe actually cooks up the final product. That 9:3:3:1 ratio, it doesn't just give us 2 or 3 options, it gives us 4 distinct coat colors. You get black. You get blue, which is really just a diluted black. You get chocolate, and you get lilac, which is just diluted chocolate. It's a perfect example of how 2 totally separate genes interact to create one beautiful, complex trait.
So if there is one thing, one single idea you take away from all of this, let it be this. The presence of a gene doesn't guarantee its expression. The simple fact that you have a gene for something is just the very, very beginning of the story.
And that leaves us with this really big, fascinating question. If our genetic blueprint is more like a set of possibilities than a strict set of instructions, and it's constantly reacting to our environment and even just pure chance, then what does a phrase like genetic destiny even mean anymore? It seems our story is a lot less written in stone and a lot more open to interpretation.